Hello, everyone. This is Minjae Kim from the Biomedical Engineering and Neuroscience Department. Today, I'm happy to discuss my works on differential phase-targeted stimulation effect on the subthalamic nucleus neurophysiology and Parkinson's disease. So as for some background, so Parkinson's disease or PD, it's a debilitating neurodegenerative disorder that results from the death of dopamine-producing cells in the substantial nigra in brainstem. So this causes an overall deficit in dopamine in the brain, which is a neurotransmitter that plays a crucial role in motion control. So specifically, dopamine binds to the receptors and this nuclei called the striatum, which then inhibits its downstream basal ganglion nuclei, the globus pallidus externus to GPE, and in the internus to GPI. But the overall lack of dopamine in PD reduces this inhibitory effect on the GP and GPI, thus hyperactivating the subthalamic nucleus in this downstream pathway, and in turn hyperinhibiting the thalamus, resulting a reduction in the output into the cortex. So the overall deficit in dopamine causes an abnormal neurophysiology across the network circuit, across the basal ganglia, the thalamus, and the cortex, that is fundamentally associated with the motor dysfunction that you see in clinic. So using pharmacological interventions or DBS, one can effectively manage the symptoms of PD by effectively normalizing this pathological neurophysiology for Parkinson's. So for our focus, the DBS, it is typically done by delivering this continuous high-frequency rectangular pulse stimulation into mainly the two basic ganglia structures that I mentioned, the STN, shown in orange, and then the GPI, which is more dorsal, shown in green. But DBS that is currently used in practice in clinic, it is done by delivering this continuous high-frequency pulse, which is then associated with the unwanted stimulation-induced side effects, including both motor and cognitive deficits. To minimize the shortcomings, we then decided to test an alternative stimulation approaches called the phase-targeted stimulations, or PTS. The mechanism behind PTS is delivering a current pulse at specific phases of a local brain activity oscillation, such as that from the SDN. Now, so effectively, PTS can disrupt the oscillation patterns and patterns of neural samples at selected phases. Now, there are several studies that have proposed the utility of PTS, but its generalized efficacy hasn't been evaluated across responses from a variety of pathological and neurophysiological markers that is observed in PD. So as per our empirical investigations, we then hypothesized that delivering a stimulus at the 180 degrees, so corresponding to the trough of the waveform, could maximally suppress these pathological or neurophysiological markers that you see in Parkinson's. Now, in order to test our preliminary hypothesis, we adopted this in silico approach. So using this neurophysiologically similar computational model to simulate the neural activity across the cortical, basal ganglia, and thalamic network that is showed in previous slide. So to stimulate this PD condition, we reduced the striatal input to the GP and GPI, just like what would occur in the actual disease state in Parkinson's. So here we then reinitialize this model such that there are about 50 neurons for each of the GPE, GPI, and STN nuclei, and recorded their membrane potential. And so we then attempted to implement our PTS paradigm. So from our recorded membrane potential of each of the STN neurons, we bandpass the signal to extract its beta band waveform. So across this beta band waveform, we then identify the specific time points at which the phases occur. So specifically, we reviewed about from the 0 degrees to the 360 degrees in 30 degrees increments. So 0 degrees, 30 degrees, 60 degrees, and so forth. And each of these time points, we deliver a rectangular pulse. Then we chose the three PD neurophysiology measures that we are interested in observing as this changes the response to PTS. The first measure we reviewed was this beta band power in the STN nuclei that are shown to be exaggerated in PD patients. Second measure we looked into was this enhanced asymmetry of the beta band waveform, which is described as this non-sinusoidal jagged triangular shape that is also observed in PD patients. And lastly, we then reviewed the degree of this phase-amplitude coupling, or PAC, between the beta band phase and then the high-frequency oscillation, the HFO amplitude. So PD patients, they also have the abnormally high frequency coupling as shown in the figure. And this is our first preliminary results, and this is a power spectrum of the beta band power across the baseline condition where no stimulation was delivered, but in the disease state, and the stimulation conditions across all phases. You can see that for baseline, we have this exaggerated beta band power in the 13 to 20 hertz range, which is replicated and then observed in PD patients. Now, for all the other PTS conditions, could effectively suppress this heightened spectral signal. But within the PTS conditions, the mean beta power in the STN was maximally suppressed across this narrow range of 180 to 210 degrees. This corresponds to the trough phase and then the rising phase of the waveform. Now, for the beta band waveform asymmetry, the degree of asymmetry was calculated using this metric called the steepness ratio, which is a ratio between the peak sharpness and the trough sharpness of the waveform. So the higher difference in peak sharpness and the trough sharpness, it will then lead to a higher steepness ratio, which corresponds to the higher degree of asymmetry. So all PTS conditions could effectively suppress this heightened sharpness ratio shown in baseline and pathological state, right? But within the PTS conditions, the 270 degrees stimulation, which is the rising phase just after the troughs, could maximally suppress the sharpness ratio. So I mentioned that the sharpness ratio is the ratio between the peak and the trough sharpness of the waveform. Now we can hopefully appreciate that in the baseline condition, the distribution between the peak and the troughs is highly divergent, leading to the higher sharpness ratio. But in the 270 degrees of PTS condition, we could see that these two distributions shift to the highly overlapping composition, effectively making the ratio closer to 1. But last but not least, the phase-amplitude coupling. So in baseline, we see the exaggerated mean pack in the SDN. But like other responses under PTS, all PTS conditions could effectively suppress the pack. And within the PTS condition itself, 180 degree stimulation corresponding to the pack, the local minima, could maximally suppress the pack as much as possible. So measure of pack can also be represented in this co-modularogram that shows a degree of signal coupling between the beta band frequency and shown in the x-axis, and then the high frequency oscillation amplitude frequency shown in the y. So in baseline, we see this exaggerated coupling streaking vertically across this 13 to 20 hertz of the phase, driving this enhanced mean pack value we've seen in the previous slide. But in the 180 degrees PTS stimulation, we could suppress this heightened cross-frequency coupling with the absence of this vertical streak. So in conclusion, our results suggest that across these neurophysiological responses, we scrutinized delivering a stimulus at a narrow range of phases about 180 to 270 degrees of SDN. Waveform was shown to maximally suppress the variety of pathological and neurophysiological signals that are measured in PD conditions. Targeting this narrow phase with stimulation may allow for motoric improvements with fewer side effects than this constant high frequency stimulations for traditional DBS. But because this is a computational modeling study, so for our next step, we need to perform on in vivo investigations on animal or human models to really validate this clinical utility of PTAs based on our findings. Now, lastly, I want to thank all my mentors for supporting me through this project. Dr. Kelly Mills is from the Department of Neurology, and Dr. Yusuf Salimpur, Paul Codella, and William Stanley Anderson is from the Department of Neurosurgery. Thank you.